next speaker, Cindy Bokobza. Yeah. So she is a perinatal neuroscientist with background in translational research, and today she will present us the results on toward effective neurotherapies for preterm brain injury, a data-driven approach to treatment optimization. So please.
Good morning, everyone. So first of all, I'm really happy to present the data here. So as you know, all of these data that we'll present you today are part of the PREMSTEM work we've done in INSEM, but one particularly is to know that half of the data that I will present was performed during the lockdown in Paris because the lab was still open. So it was a funny period for Pierre and I in the lab. So just a quick remember, but I know that every one of you here know what is preterm birth. So it's any birth occurring before 37 of complete gestational week. It represents almost 50 million of infants every year. And the main lesions that we observe of white matter injuries, so historically they were necrotic lesions, but now we observe more diffuse and subtle injuries. But what we know is that even if there is advance in IQ, preterm infants have long-term consequences of these injuries, including neurodevelopmental disorders. And as a societal cost, it was estimated that the perinatal care, short and long term, represent almost $14 billion every year in the US. So if we go down now to the cellular level, what we know both in human and mouse, and I will go back to that afterward, is that the glial cell are really at the heart of preterm brain injuries, especially oligodendrocytes that are the main producer of myelin, and microglia that are the immune cells of the brain. So here I show you some postmortem histology from 33 gestational week preterm infant, and here, as you can see around the lesion here, if we zoom in right here, we can see that around the lesion we find glial cell, both astrocytes and microglia. So I will not redo the amazing keynote that we had yesterday about stem cell and how we use them to repair the brain. But as you know, they are really good to do to have anti-inflammatory properties and repairing the brain. And during the PREMSTEM project, the idea was to use for all the members of the consortium the same stem cells that was furnished by Chiesi, that was umbilical cord-derived mesenchymal stem cells. So just keep in mind that at the beginning of the PREMSTEM project, the idea was that we want to have one stem cells that rule all the problem and that can resolve all of the injuries. That was the hypothesis at the beginning. We will see if it works or not. But the first step was to find a way to determine which was the best paradigm to inject this stem cell to the animal model, both in rat and in sheep, for example. But first thing first, I'm going to present to you the mouse model that we have in the lab and characterized for more than 13 years. So way before I arrived in the lab. So basically, it consists of intraperitoneal injection of the pro-inflammatory cytokine interleukin-1 beta, the first day of life of the animal. Why? Because this period corresponding to the brain development in mouse of the third trimester of pregnancy in human. What we know in the mouse model is that IL-1 beta induces an oligodendrocyte maturation deficits that we can observe as soon as P15 and confirm at P30. And when we look at one cause of why we have this lesion, we went directly to microglia because we know it's glial cells that I show is surrounding the lesion in a human. So what we did is that we inject gadolinium that will deplete selectively pro-inflammatory microglia, and when we do that in our model with IL-1 beta, we can see we have a reduction of the white matter injuries, demonstrating that microglia is really the cell that is responsible of the onset of the lesion. But PREMSTEM is not about mouse, it's about rats. So the first thing that we did was to transpose this mouse model to rats and confirm that with IL-1 beta we were still able to see in rats the lesions that we are perfectly expert on in mice. So what we did in PREMSTEM is that we still inject from P1 to P5 interleukin-1 beta, but we had to double the doses of IL-1 beta than in mouse. So first, on the general animal body weight, we were seeing that, as in mouse, IL-1 beta induce a reduced body weight gain over time. And when we cell-sorted microglia, so CD11BC positive cells at P3, we were able to see by qPCRs that we had an overexpression of pro-inflammatory gene at P3, and also that microglia were releasing way more reactive oxygen species in rats, coherent with what we saw in mouse. We also perform microglia cell sorting RNA-seq analysis. So as you can see here, it's a global heat map, but as in the mouse, what we saw is that we had the cluster A and cluster B that demonstrate a downregulation of the developmental function of microglia to an overexpression of pro-inflammatory cytokine by microglia. What was interesting in this data set in rat is that we demonstrate that there were a specific cluster that was still there 3 weeks post-inflammation that was also overexpressing inflammatory gene. So there is still inflammation going on 3 weeks after the last, 2 weeks, sorry, after the last IL-1 beta injection. We then also confirmed that we found in rat model oligodendrocyte maturation delay. So first thing first, we did again RNA-seq sequencing at 3 postnatal week, and what we saw is that by IL-1 beta oligodendrocyte, we have a downregulation of a pathway associated with oligodendrocyte differentiation, and we have an upregulation of pathway that negatively regulates oligodendrocyte maturation, which was coherent. Another way to demonstrate it is that we, in France from ICM, there is a database that gives what is called an oligo score. So basically, if you give them the genes that you find in your model, they will tell you if this gene in oligodendrocytes are associated with which function of oligodendrocytes. So you have myelination, differentiation, survival, migration, proliferation. And here is the data from IL-1 beta, and as you can see, we have a low to a negative score in regard of myelination and differentiation in oligodendrocytes from animals that was treated with IL-1 beta. This was also confirmed by histopathology because here, as you can see, in IL-1 beta, we have a reduce of MBP density in the corpus callosum. So now, first steps that we did in PREMSTEM. So the idea, as I said, was to find the better paradigm to inject the stem cells in our model. So we had an amazing, fancy idea. We compare something that was never done before, so 18 paradigm. So let me get through you this 18 paradigm. So first, we have the stem cells that were provided by Chiesi, and we wanted to test it at 3 doses, so 20 million, 50 million, and 125 million of cell per kilogram. Also, we compare 2 routes of administration, intranasal versus intravenous. So as I said, the last injection of IL-1 beta is at P5, so we decide to test it 3 age of administration of the stem cells, so P5, P10, and P20. And for all of this comparison, we perform microglia transcriptomic analysis 2 days after stem cell administration and MBP quantification 10 days after stem cell quantification. And because PREMSTEM is not that easy, we go even further because from the best paradigm that was analyzed from the IL-1 beta model, we confirm in another perinatal brain injury model that was done in Gunberg with germinal matrix hemorrhage at P5 that we also rescue the brain lesion due to preterm birth. So as I said, we confirmed 18 paradigm, 3 doses, 3 time points, and 2 ways of administration. So how did we do it? So the first thing first was looking at microglia transcriptomic. So we did with bioinformaticians, so collaboration with the team of André Delahaye in our lab. So basically, I'm not a mathematician, so I will try to explain it to you as best as I can. So we look the transcriptomic profile of microglia treated, like injected with IL-1 beta and then treated with MSC, and we look for each gene how much this transcriptomic look like to the control, so PBS. And this was giving through a score going to -1 to 1. So if you have a score that it's close to 1, that means that this paradigm is a good candidate for treatment because it's closer from the control microglia. However, if you have a score that is close to -1, that means that you are mimicking the disease and it's not a good candidate for treatment. So here are the results for our 18 paradigm in the IL-1 beta model. So the 2 take-home message from this table is that first, as you can see, we have a better score from the intranasal administration in comparison to the intravenous administration, and also late administration around P10 and P20 seems to be better at transcriptomic level than early time points. And then we confirmed this 10-day post-administration by looking at MBP expression in the brain, so we did it by Western blot, and if we saw that for approximately all of the condition, back to normal expression of MBP, what was striking is that here, as you can see at P20, the intranasal administration of stem cells seems to overexpress MBP after treatment and the stem cell administration, which was surprising. We not even only resolve, we also ameliorate the brain. Another way to present it here is just the score in abscess and the score towards the MVP. And as we can see here again, the best paradigm was the P20 administration at 50 million of cells per kilogram in the IL-1 beta model. Now go back to the double confirmation in the other model of PBI. So they compare several paradigms at P10 and P20, but here I'm presenting only the data from the P20 50 million intranasal administration. So here is MBP expression in the cortex, and as you can see, with the GMH, we have a down expression of MBP in the cortex of animal, but with the best paradigms that we demonstrated in the IL-1 beta model, also in the GMH model, we were able to restore MBP expression in the cortex of animals. What is interesting to say is that the best paradigm was determined by transcriptomic profile in one model, and we didn't have to do again this transcriptomic profile in the new model, which is demonstrating that the scoring and the screening is efficient to all of the PBI model, hopefully. So now if you go back more deeper in the IL-1 beta model, I just analyzed the pathway associated with MSC both in microglia and in oligodendrocytes. So we had a lot of genes that were dysregulated by MSC in comparison on IL-1 beta only model. And as you can see, what the MSC was doing at transcriptomic level 3 post-anal week, it was mostly downregulated inflammation. So it was a specific clusters that I talked to you earlier on microglia and also on oligodendrocyte. There is this signature of inflammation that's going on at 3 post-anal week, and that's what the MSC is working on. Also, we confirm that the myelination is restored by electron microscopy. So as you can see, in IL-1 beta only treated animal, we have a reduction of the J ratio on the corpus callosum that was restored by MSC treatment. And also, we perform behavior analysis and brain structure analysis by MRI. So here it's the result from open field tests. So as you can see, in rats, IL-1 beta induce an increase of locomotor activity from animals that was restored by the treatment with MSC. We also perform YMS tests to evaluate the working memory, and if we have deficit of working memory induced by IL-1 beta, again, it was restored by the treatment of HMSC. On this same animal, at the end, we collect the brain and perform MRI analysis. And as you can see, in several regions, we have microstructural brain alteration in several brain regions, such as the corpus callosum, the external capsule, and the motor cortex. And it was, again, restored by the treatment with HUMSC. In conclusion, first, we developed a robust and scalable rat model of preterm brain injuries aligned with the human pathologies. We use an unmatched experimental firepower by testing 18 MSC delivery paradigm and identify late intranasal administration as a top performing strategies. And what was interesting is that this scoring of this late intranasal administration was also translated in the second independent model injuries, which was GMH, without any additional transcriptomic profiling. If we go back to IL-1 beta, we demonstrate that HUMSC treated animal recovered not only at the molecular level, but also structurally by electron microscopy and MRI, and also behaviorally. So in conclusion, our data define an optimized MSC protocol and a reproducible framework to bring it to the clinic. So to conclude, I want to thank the PREMSTEM Consortium. It was my pleasure to work with you for the last 5 years. It's kind of a bittersweet for me right now because it's the end, but I'm sure we will keep collaborate all together. So thank you very much, and I'll take any question.
Thank you. I think that we have time for one, max 2 questions.
Congratulations on an enormous amount of work. That just looks, it's very impressive. I wonder if you can comment, please, on your dose response for your outcomes related to, I think, myelination and your overexpression of myelination. Were they then, could you relate them to any functional indices, and can you relate that dose to what should be used in the large animal models and potentially then in clinical trial?
So in regard of why we have this hypermyelination, so I was this result was the reason why we perform oligodendrocyte transcriptomic analysis, because I was really curious to understand what's going on in this oligodendrocyte to overproduce myelin. And what was interesting is that there were a specific pathway that right now undercover in other myelinitis disease that seems to be restored or stimulated by the MSC. So this is something we want to go deeper. But now, and also what is interesting is that we have to correlate this data in regard of electron microscopy, because if you look at the image, the truth is the axon inside the corpus callosum are not the same as the control. Like, it's mostly affected the big axon, and we can see this hypermyelination by the density of the myelin around the axon. So there is definitely something we want to go deeper right now, but it's already a big thing in the oligodendrocyte field. So with Pierre, we are questioning on that right now. And in regard of the dose response to large animal model, the thing is right now, as it was discussed during Sarah's talk, like, we don't know exactly how many stem cells going inside the brain by intranasal. So probably in large animal, we do not need that much. And because intravenous will work better in large animal because it's easier than intranasal. And that was the thing that was demonstrated in team lab during the control.
the last one.
Thank you so much for the comprehensive analysis you've presented. I was just wondering, on all of your first analysis looking at, I think you were analysing 2 days after each of the doses. Can you tell us a little bit about the timing for the later studies where you were looking at behaviour?
Mm-hmm.
How long were those behaviour and neuropathology after the cell doses?
So basically, all of what we try to do is that, so on the scoring, it was 10 days, so approximately 1 month old animal in rats. And we tried for behavior to not go up to 2 months and a half. So it was a lot of logistical paradigm, but we tried that to finish all of behavior before 3 months old of animal. So we were still in the coherent period in regard of what we demonstrate from the scoring. And also it was true for electron microscopy. Everything was tried to be all in the same period of time.
Thank you very much, Cindy.